we're bang on time. So how much time do you want to allow us for questions? 5 minutes? Yeah, 5 minutes. Okay. So I'm going to allow 3 or 4 burning questions, short questions that is, and short answers, please. So somebody up here at the front in the pink. I think it's pink, unless I'm colorblind.
My name is Irina Sidorova. I'm a participant of Biobank, and I'm a supplier of data, and I'm a consumer of data. And 3 years ago, Labour government decided to create new initiative and create patient research ambassadors. I would like to ask if your research have a public and patient involvement or patient research advisor, because it's such a nice event. It's about 500,000 people, 50,000, something like that. But I would like to see my liver, my brain. I would like to participate on the research, what is related to me, and probably deliver findings to my community. Because sometimes you guys, you so enjoyed that research. That is a huge potential or something like that. Why we cannot stick with a lifestyle, like to be vegetarian, quick smoking or drinking
Can
or
I
something like. Yeah, I'm finished.
Okay,
Thank you.
no, it's okay. No, I think it's a good point, and I think UK Biobank. I mean, maybe Andrew, you want to take that because we have had participant, meetings where we have participants. Maybe Kathy,
Yeah, so we do involve participants as best we can. We have participant meetings. We encourage them to make contact with us and talk to us about how the resource can be used. But
no presenters, 3 years initiative and nobody. This everything right.
Okay. Yeah. I should say my mother-in-law is also a participant. So
I do think it's a very good point, and actually the previous few meetings we've had have involved participants in the same meeting as the scientists, and in some respects that's been very successful, but in other respects it's brought challenges. So we're constantly evolving and experimenting, and we intend in the future to run a series of different types of events, meetings for scientists where science can be discussed perhaps in a way that's not accessible to the vast majority of participants. It will be to some, like yourself, and that's why you're here, but perhaps not to all. And there may well be, so you've suggested something that's perhaps a seed, which there may be people who are participants and scientists, and we know some of them already. In fact, there have been some on the platform, but they haven't maybe self-confessed that. But who can plug exactly that gap? So maybe it's happening partly in silence, but perhaps we should be more explicit about it in some of our events. But we do a lot of this.
Okay, thank you for your question. There's somebody up there in the middle there. Again, is that a pink shirt? No. It's not quite pink, actually, but it's close enough.
Okay, Ron Unger from Bar-Ilan University in Israel. All of this new omics stuff, compared to the genomic, the DNA level, it's one time short. This thing, all of the proteomics and metabolomics are changing during lifetime. So do you plan, or the idea is to do it every few years, every few whatever? Yeah,
so I think each of you can quick short answers. It's a moving field is probably the question, isn't it? So Adam?
Yeah, that would be great. I think the first challenge is do it in Biobank at all, but then the fact that there are some serial blood samples taken from people who've come back for other imaging, etc., then yeah, I think that would be great to do.
Ellen, do you want to, sorry, we'll go with, sorry, Peter.
Yeah, so maybe for DNA methylation or epigenetics, a little different. I think we'll be excited to see at one time point could that predict future outcomes. So maybe proteomics, metabolomics, it's more about, for epigenomics, I think we're talking about one time point, but rather see how could that predict future outcome.
Yeah, and Peter?
Yeah, it's a good point. There's a lot of value of assessing the metabolic profiling at multiple time points. So as part of the initiative with UK Biobank, we will be measuring around 20,000 samples that are collected and a reassessment. But then I think that also other biobank studies can complement the information that is gained from UK Biobank, which is largely a single time point. Then other studies have reassessment multiple times. So it's not UK Biobank will give the whole answer. Other studies remain important.
Okay. Anybody have one last burning question? Okay. John Todd, young John Todd.
Yeah, the price that Nightingale and Biobank negotiated must be very competitive for 500,000 people. Does that mean we can all enjoy that price?
Peter, you want to disclose that or not? I mean, it's up to you entirely.
So there's a press release out this morning with details some of these aspects. So this the biomarker profiling in this case is fully funded by Nightingale, and eventually the data will be released for the biomedical research community. But in the same way as the exome data, there will be an exclusivity period before the release. Yeah,
and I think as the committee, and I'm involved in the enhancements committee, we're looking at other platforms clearly, as we've already heard. But in terms of the robustness and also when they're value for money, and I think those discussions are ongoing. But I think we're encouraged from the start you've seen so far. Going forward, I think we will bring on more platforms to enrich this resource, but we're just taking our time, make sure we get it right. I think that's the key. Okay, I think we'll stop there. I think we all need a bit of caffeine. Thank you for the 3 young speakers, and see you this afternoon.